Maybe I move away from Tomas to be better. Yes, so today I'm going to talk a bit about what my research group and what we're trying to do at RMIT is working with for studying stem cells and trying to track temporal changes in them. So maybe you can understand why I'm very keen on the fact that we've just got our FluidFM installed this year and we can start doing experiments, but no, there will be no FluidFM data in this, but that's okay. So before I do get started in the talk, I do just kind of want to do something that's very typically done in Australia. So I've been walking around Zurich. I love being in Europe. It's amazing. It's so old. It feels so old compared to Australia. So, you know, Zurich was established, I think, as a Roman town 2,000 years ago. You know, there are cobblestones that are older than any building in Australia. It's fantastic. But Australia was probably colonized about 250 years ago, but it has been inhabited by the First Nations people for about 65,000 years. So in Australia, when we present our work, when we give meetings, that kind of things, we always do what's called an acknowledgement of country. So today I'd like to share with you an acknowledgement of country. So RMIT acknowledges the people of the Woiwurrung and Boonwurrung language groups of the Eastern Kulin Nations on whose unceded lands we conduct our business on. So all the work presented here today has been done on these lands. And so we like to respectfully acknowledge the elders, past, present and emerging. And in general, we always acknowledge the traditional custodians of the land and the ancestors of the lands and waters across Australia that people from Australia we do our research in. So this is just something that we always like to do, and because I'm presenting work that is being done on this land, even though I'm not on country today, we are just acknowledging them as well. So to kind of give a bit of background into what my research group is doing is we are kind of generally in the sphere of using stem cells for personalised tissue engineering. The idea that you can take stem cells from a patient, from an adult patient, from their bone marrow, from their adipose tissue, from their dental pulp. You can isolate mesenchymal stem cells. I'm not sure if the lasers... Oh, there it goes. You can isolate mesenchymal stem cells. So these are kind of, they'll go into osteochondral tissue, so your bone, your cartilage, muscle, sometimes neuronal, depends who you ask. But generally they're quite good when it comes for tissue engineering applications, because the idea is you take the patient's own stem cells, you isolate them, you grow them, you push them towards the targeted phenotype that you want, whether that's bone, fat or cartilage, for example, they're the common ones. You can grow new tissues, you can either inject them directly or you can put them into a biomaterial, some kind of construct, reintegrate it back into the body, repair or regenerate that tissue. And because it's a patient's own stem cells, the idea is that you should reduce the effect of graft versus host disease. Because ideally the patient's body is like, hey, come back guys, yeah, it's all good, you're one of us. And hopefully the cycle completes and the patient ends up in a much better place. And so what my research is specifically looking at is, because I'm not a biologist or an engineer, so we're looking really at this little part of the cycle here. So how do we manipulate those stem cells to turn into the tissue type we want? Can we do it more efficiently? Can we create techniques that can maybe use the same bioreactor to push cells to different phenotype outputs? And so looking at how we can manipulate stem cell response and promote different outcomes at this stage here is where I really kind of fit in. And so in the world of biomaterials and tissue engineering, which is obviously a very big field, most of this is done through engineering the microenvironment. So there's the classic Engler paper that came out showing that if you grow mesenchymal stem cells on substrates of different stiffnesses, so very, very soft materials, you will get neuronal-like cells. Very stiff materials, you will get bone-like cells. Cells respond to that microenvironment. It's all about how they create tension and how they spread and the things that get activated within cells when they do have a high tension. There's a lot of really good work being done on nanotopography, so changing the topography of surfaces by making cells stretch and grow and align to things. You can push differentiation, and this can be all done without any exogenous factors. We're not providing growth factors for this. The cells are just responding to this microenvironment. And then there's also a lot of work, especially in the US, done with chemical controls, providing different chemical ligands, different kind of hydrogel networks, patterning, that kind of thing. Again, using that to push cells down specific tracks, push them towards certain phenotypic outcomes. Oh, one ray. And most of that is due to the fact that these stem cells are very mechanically sensitive. There are a whole raft of different pathways on how cells will interact and respond to their microenvironment and form tension from Things just like the surface topography, substrate stiffness, and then how the actin cytoskeleton develops within the cell, how that links up through to the nuclear envelope. There was a great talk yesterday talking about like chromatins and how the nuclear envelope and how things attached to that help drive differentiation outcomes and gene changes in the cells. And so it's a very common way to kind of use biomaterials to kind of regulate stem cell outcomes. So like I said, different stiffnesses, you can even pattern depending if you can like disrupt or control the cell cytoskeleton, that intracellular tension. That's really the key that we see a lot of this work. And then you'll get all these different kind of phenotypical outcomes from that purely by just controlling intercellular tension, cytoskeletons, and how the cells create tension like that. So this is kind of where I came into the field, and while it's great you can create like all these biomaterials, there are groups out there that do a lot of work in developing like the perfect biomaterial to support bone differentiation and to help bone reintegration in the body, or neurons or other things. And so because I'm not a biologist, I'm like, why do we have to limit ourselves? Why can't we just try to fix all of it? Why can't we get as patient stem cells and get the multiple outcomes out with a single kind of system? Because if we use external stimulation, so rather than a passive biomaterial surface, we are providing a cue externally, we have a lot more control over that. We have both temporal precision, you can have some spatial precision depending on the way that you provide this stimuli. We just saw in the previous talk, pushing down on cells, you can get a response from that, you can control that spatially. And so if we can kind of manipulate this pathway from stem cells to specific phenotypes with the external stimulation, we kind of open up a whole different can of worms, but hopefully good can of worms, with the idea that we can condition these cells, we can precondition them. I haven't talked about it too much in these slides, but there's this whole kind of area of stem cell research going into mechanical dosing, because stem cells have a mechanical memory, and if you do something to them that actually affects their mechanical properties, their biomechanics, you can kind of condition them, and later on they'll still turn into the stem cell that you want, depending on that preconditioning, because they do seem to retain memory of what you did to them. Did you push them through confinement? Did you stretch them? Did you squeeze them? Even if they kind of revert a little bit later, they'll still remember that. And it can be really useful if you want to pre-treat your cells at this phase and then integrate them into different kinds of biomaterials or constructs without needing that carefully designed biomaterial that will only promote one outcome. So that's kind of where we're at. We're looking at external stimulation. And so what kind of stimulation? So my background through my PhD and a first postdoc, I worked a lot with conductive biomaterials, namely polypyrrole, which is a conductive polymer. It was kind of fun because it's see if the video will play. I think I might need a press play on it. Let's see. Oh, there it goes. So we made electroactive scaffolds that they will actuate when you apply an electrical potential. It's kind of a fun part of conductive polymers. You can also change their surface properties and switch them and change them. And so when I started doing kind of more work and I went to Imperial College and we were looking at how you can drive differentiation with stem cells with electrical stimulation, the more I looked into it, the more it seemed to be clear that we don't actually really know how the cells take this charge injection from a conductive material and transduce that into a biological signal. And that's kind of what the postdoc in Imperial was trying to explore. And so we know that there are a lot of different mechanisms that can be affected by electrical stimulation. Nobody's really clear that yet in the field of what's happening with the stem cells. So we know that if we electrically stimulate stem cells, we can get cartilage differentiation, we get bone differentiation, smooth muscle differentiation, and again, some neural-like cells. But nobody's really clear on the mechanisms as to why this happens. Papers publish one outcome, they maybe look at a couple of genes, if that, they're like, yep, cool. That worked, but a lot of times there's no real rhyme or reason as to the stimulation protocols that they use. They can be very hit or miss. You don't know what kind of charges they're putting in. You don't know what kind of materials. Sometimes nobody really quantifies the biophysics side of the charge injection into the cell, and conversely, there's a million different permutations that you can look at. You can look at sinusoidal waves, triangular, pulses, biphasic, consistent, like do you have an electrical field or is it just charge injection? There's like a whole raft of different settings, and it's very unclear as to what's actually going on. And so there could be a few things here. It could be ionic gradients around the cell in the media, especially with electrical fields. It can be, obviously, you've got your voltage-gated ion channels on the membrane of the cells, and membrane potential. Again, that's obviously going to be very sensitive to any kind of electrical stimuli. Calcium flux is something that people are kind of thinking of. Calcium is obviously involved in many important kind of pathways within the cells. If you are triggering calcium flux, but either internally or having release of calcium, you may be triggering different pathways of the cells there. Cytoskeletal rearrangement, as I said, it can be very important for stem cell differentiation, and then also just kind of electrophoretic rearrangement of surface molecules, receptors, as well as extracellular matrix deposition around the cell. So these are all things that people have kind of pointed at, and we're not really sure yet. Lipid rafts are another one. So it's very unclear. And the reason why no one's really figured out what of these parameters are affecting it is because it's really hard to study. Again, if you want to study a couple of variables and you want to try to see if the cells are changing at all, it's a very complicated experiment. I think at Imperial we looked at like 2 or 3 different stimulation protocols, and 2.5 years later we had like maybe some results, if that. And you can't really buy stimulation devices to do this. You have to make them yourselves, and so the amount of PDMS and copper tape that I've gone through in my career, hopefully never do that again. So we've got some lot more sophisticated devices now that we've made, because at RMIT we have fantastic fabrication facilities, but it's been an uphill battle. So the question that we're really trying to answer is, well, we know that cytoskeleton intracellular tension will affect stem cell fate. That's through biomaterials. We know that's going to happen. Substrate stiffness, topography, all that kind of things. Absolutely. So we know electrical stimulation will affect stem cell fate. We can drive it that way. So kind of the question that I ask is there a way that electrical stimulation is affecting these mechanotransduction behaviors of the cell? Is there something going on here where we're actually affecting these cytoskeleton structure, intracellular tension, focal adhesions? Is that how electrical stimulation is having an effect here? But one of the challenges in this is trying to capture that point of charge injection. What is happening when the cells are feeling the stimulation? So today I'm going to talk mostly about just the polypyrrole, but we do have some other materials that we're looking at, and on the biophysics side of things, I'm very curious about materials with different electronic properties. Polypyrrole is kind of what we call a pseudo-capacitive material. So there's ionic charge through the backbone of the polymer, and then it injects charge that way. It's very safe and biocompatible. We don't get any unwanted byproducts with electrical stimulation. We're also working with some nanodiamond, which generates a photocurrent if you shine light on it, which is also easy to set up experimentally. And we've also trying to do some work with things like ITO glass and graphene and metals and that kind of thing to see if there's a difference with the electronic properties, but not today. So a classic picture. At Imperial, we had a very basic AFM. I don't know if you know the VTech ramens, they have an AFM adaptation to that. That's what I was working with. It's fun. It's really fun. And so we had live cells, and the plan was to indent our cells and look at the Young's modulus before and after stimulation. And so with the setup we had, we could measure individual cells once pre-stimulation, which was all these black points here. Then we would take the dish away and stimulate in the incubator for an hour, and then bring it back. And so this is kind of just one of the experiments. So this dashed line here represents where the stimulation was applied for an hour, brought the cells back, and then indented a whole bunch of cells over time. And so we could see that generally the cell population was getting softer post-stimulation compared to doing the same thing, but not doing any stimulation, so we know the cells weren't just dying from the treatment. But it was pretty slow. We had to use colloidal probes, so we were only just getting the general indentation of the cell. We had, again, great talk just 2 talks earlier talking about, this is a very simplified way of looking at the cells. They are not just big homogeneous balloons of water. There's a lot of things going on there. So going to RMIT was fantastic because we had a JPK NanoWizard 4, which was newly bought, not for me, just happened to be there when I rocked up. That was great. And so we could actually use the QI mode. And so if some of you may have a NanoWizard or not, but they're really handy because instead of doing force spectroscopy, or even force volume maps, which are very time-consuming, QI mode is kind of like tapping mode, except every tap is a force indentation measurement, but it does it at the speed of tapping mode. But you are very, very gentle. There's not really any kind of lateral force on the cells. You come down, you push, you lift up, and then you go across again. So it's more like hopping, kind of like SICM even. So it's really fantastic for imaging live cells. So this is one of our stem cells. So you get a really lovely topography image where you can really clearly see the cytoskeleton structure. It works fantastically with the human mesenchymal stem cells because they're nice and flat. So we see beautiful pictures. So you can get the stiffness of this, and you can also do adhesion. And so I'm not going to talk about it today, but one of the plans as well is to do functional probe scanning, where we functionalize the probe with a specific target, and then we look for something on the sample and on the cell surface and look at unbinding forces. But not talking about that today, just talking about indentation. And so it's really nice because now all of a sudden we can get very high resolution images of the entire cells, 65,000 force curves, and really start to understand maybe how those intracellular changes are happening with stimulation. So we started out obviously with controlled experiments where we could change the cytoskeletal structure of the cells by using chemical disruptors. So this is cytoclastin D. You put it in, it really messes up the cell, dissolves all of its actin structure, gets very sad and soft. But then over time, it begins to build up and rebuild its cytoskeleton structure. And so we can see that here, that these again, like 65,000 force curves of a cell, you can clearly see the mean stiffness of the cell drop and then begin to rebuild back again once we take out the disruptor. And so we've also done this with blebistatin and negadozole and colchicine as well. So different disruptors that affect different parts of the actin cytoskeleton. But what's good is now we can kind of do these measurements, but we can do them on these little homemade devices where we can apply stimulation while the cells are growing on the chips and do these biomechanical measurements at the same time. That's what I said. So this is, Kaiwen likes to call this the frog cell because it kind of looks like a frog, which is quite fun because it's, if you throw your mind back all the way to maybe your early physics days, like thinking about what's his name, Galvin? I know, like Galvin of Texas, so the guy who did all the experiments on the frog and light would electrocute them. It's a little bit like that. So we start our pre-stimulation, we can see the topography of our cell, then we keep imaging it during electrical stimulation for 1 hour, and then we continue doing that for several hours post-stimulation. So this is just a few select cells. So here we've got the Young's modulus. So the Young's modulus data that I'm showing here is not from the JPK software, it's from external Python code that was written by Joe Berry, who's a collaborator of ours at University of Melbourne. And so you can clearly see the stiffness of the cell is changing here with the electrical stimulation. You can really start to see the stiffness building up in certain areas of the cell. It's probably a little bit difficult to see on the slide here, but when you look at it up close on my computer screen, you can also see changes in the cytoskeleton structure as well. And so this is one of our first results. We're really excited because you could very much see that the cell is responding and changing to the electrical stimulation as compared to our control experiments where the cells just kind of look the same. Sometimes they move around a little bit, but they're not really that active. So we also can, when Kaiwen's doing these images, he goes and zooms in on different parts of the cell as well. And we can see changes in the Philippedia, and then he calls them the little hands as the cells kind of change and respond to electrical stimulation. Again, I don't have the data on these slides, but we've also kind of just imaged the polypearl substrate that they're growing on to ensure that we're not getting any changes on the surface itself with the electrical stimulation. And these are very fast pulses. They're 100 millisecond pulses at different voltages. So that works out really nicely. So we do our lowest charge, which is 0.005 volts. Like I said, it's 1 pulse per second, 125 milliseconds. It's a positive and then a negative charge so that it neutralizes the charge and we don't get charge build up on our substrates because we work without a reference electrode. It's a situ electrode system. We see the low ones, the cells kind of go all over the place. Sometimes they get stiffer, sometimes they get a bit softer, and then we have the control cells without any stimulation just kind of hanging out in the middle. And so this is a whole cell average of about 10,000 curves, depending on how big the scans were that he did. But when we do our higher charges, when we take up to 0.5 volts, we really see that softening. So it was really nice to see that what I did in Imperial was true. That's always nice to see, but we can have a look at that and see that on average our relative Young's modulus is dropping with stimulation. That's not supposed to be there. Imagine that's supposed to be over there. It's just hopped its way over. But when you look at those 10,000 data points of the cell, and we've done it so that we don't capture any of the underlying, the external substrate, it's only the cell values from our scans, you can clearly see the mean of the cell stiffness is also dropping up, and you can see in these violin plots that all the data points are pushing down towards the softer scan. So that was really nice to see there. So we can really see that with the biomechanical analysis, we're clearly getting changes in the cells. We're seeing this change in the Young's modulus of the cells and their cytoskeletal structure. We do quite a lot of correlative, regular kind of biological analysis with immunofluorescent staining and that kind of thing, but it's quite interesting because I've been doing a lot of imaging of things like focal adhesions, actin, the lipid rafts, so looking at caveolin and cholesterol lipid rafts in the cells, as well as phosphomycin light chain, which tells us when actin is activated or not, and even YAP, which is an intracellular tension, and nothing is really as conclusive as the AFM, so it's really capturing that transient change. And we know that this 1 hour of stimulation is not really enough to drive differentiation long-term. You usually need to do like an hour a day for 5 days to really start to push that, but it's really interesting that the cells are responding to it some way. But it's a little bit of the chicken and the egg problem. We're not really sure what's being triggered. Are we affecting the focal adhesions of where the cells are, and that's making them kind of soften, because if they're losing focal adhesions, then the actin's going to depolymerize while it tries to figure out the next place to bind to, or are we doing something like affecting the calcium signaling inside the cell? So there's a lot of questions still as to what is behind this softening here, and how do we capture that? Because again, this happens within an hour of stimulation, and then it happens for a few hours after stimulation of the cells, but then within 24 hours, they're kind of back to normal. That's what's going on. So that's why we got the FluidFM. I was very excited kind of when I saw those first papers come out, and I was like, oh, this is amazing, and couple this AFM. So now we can couple our biomechanical analysis with biochemical analysis, because that will kind of help us understand what's going on. The big project, the reason why we got it, what was in the grant that we really want to try, is doing that temporal kind of, the single cell life-seq. If we don't know what we're looking for, that's the problem. We see different genes people say are important or not with electrical stimulation, but I'm kind of taking the shotgun approach of, well, let's just look at everything and maybe we'll find something that nobody has seen yet. So if we do a whole transcriptomal analysis, then maybe we will begin to understand what is being triggered here, what's going on, what's happening with the cells. If we try to pick target genes by doing, say, PCR, then we're probably going to miss something along the way because we're still not sure what we're looking for. So the idea is that we will do exactly what we've done with our biomechanical analyses. We can stimulate our cells, and we can take single cell biopsies over a period of time and see what's changing with stimulation. So that's very exciting. We are also hoping to do some nanoparticle uptake studies with collaborators. We do work with cubosomes, liposomes, nanodiamonds. That'll be pretty cool. That's like a side project. It's not really my thing, but they're interested in it, so that's all good. And patch clamping, which I hear is the bane of some people's existence, but again, normal patch clamping, the stem cells are usually too thin, so it's very difficult to do, but the idea, what we would really like to do is try to do some patch clamping, or at least the patch clamping idea, but then you're measuring what is being transferred through the cell, because again, in a biophysics kind of approach, we don't know how the cell transduces that electrical charge signal. Is the cell membrane resistive enough that it doesn't even get in there? We're just affecting the cell membrane, or is charge going to be passed through the cell? And could we measure what's passing through the cell as part of the circuit if we're patched onto it and then apply stimulation from underneath? So we're not sure yet, but that is another kind of crazy idea that I've had that I would like to try, but we'll see. And I will just end up, because I also know that we cannot miss axe throwing, so I'll finish up. If you think about potential postdoc projects, or a PhD, but I think everyone here has at least started their PhD, within Australia there's a couple of postdoctoral fellowships that you could apply for. They're both more independent positions that you apply with a supervisor, but you get to do your own thing. There's the ARC, Australian Research Council Early Career Fellowships, which are called DECRAs. They're 3 years with salary and research funds. Or there's RMIT itself has some specialised postdoctoral fellowships, again 3 years with salary, research and funds. If you would like to know more, come and chat to me about that. Because in Melbourne we have beaches, we have bridges over rivers, they're beautiful. We have trams, just like you guys. We have axe throwing, we did that last year. And we legitimately have the best coffee in the world. That's not just me saying it, we've won awards. Melbourne is a very livable city, it's very beautiful. I haven't got any pictures of koalas or wombats or echidnas, but I have seen them. And I've talked to enough people that I would not put up photos of some of our other wildlife that people get a bit worried about. I have to say I've never seen a snake in Melbourne, so you'll be fine. But Melbourne is a very beautiful place, and now we have a FluidFM there. I hoped maybe people might be interested in coming down. Check it out, check us out. We're only 26 hours away. That's great. You'll be fine. Yeah, so in summary, we have seen that electrical stimulation will induce transient changes in stem cells, but we still don't know what is going on. It's very difficult to pinpoint. AFM, obviously, as we all know, is fantastic for this temporal analysis, and I keep bringing that back to all of my biomaterials and tissue engineering conferences, and people are getting more and more on board. Every time I show up to a conference, I'm always the AFM person, so I feel a little niche, and then I was like, guess what? I'm going to an even more niche place now. I'm going to do FluidFM, and no one else does that in Australia, so I keep digging myself into an isolated hole down there, but that's fine. You may have noticed as well that the polymers we use are black. Not great for FluidFM because we can't see the cells. So all that information was my student Kaiwen going down and kind of blindly searching around for cells until he finds them. So that was a struggle itself. But we are having a collaboration with Marcella Bilek at the University of Sydney to move to transparent electrodes, which will be fantastic. So with that, acknowledgements of Kaiwen. He was the one who did most of the AFM, if not all of it, that we saw, as well as the rest of the group, and Joe Berry, who did our specialised modulus measurement analysis, and the Australian Research Council for the Discovery Project, which has funded all this research. So thank you. I'll take any questions.